In this video, we'll be talking about the rDNA and its products. That means after achieving the rDNA, what are the desirable proteins and how to obtain them. That is why recombinant technology is very important, because it helps us to obtain the proteins which are desired by us or are required by us for various purposes. So if we talk some of the recombinant proteins which are therapeutics, that means which are used in treating or is therapeutic in nature, are DNase 1, antithrombin 3, insulin, interferon A is it, A. So there are many which are being produced by this technology. Tissue plasminogen activator, many are there. Now basically we have studied that rDNA is transferred into the host cell. It is a gist we are talking of, that rDNA is transferred into the host cell where it multiplies in suitable conditions to produce the desirable proteins. Now these desirable proteins are known as recombinant proteins. So we can define the recombinant protein as the proteins which are being produced by the expression of recombinant DNA in a heterologous host. This is very important because the host is important where it multiplies, and it is different from the original DNA, the fragment from which organism we have obtained. So it is completely different. So therefore heterologous host. Now once the DNA, that is the recombinant DNA, is transferred into the host cell, it gets incorporated into the genome of the host cell and starts multiplying and will express itself under suitable conditions. Now, as told, the important aim is to achieve the desirable proteins or the recombinant proteins. Now, for this, obtaining the desirable protein, that is the foreign gene product, it can be produced in a small scale or it can be a large scale production. Two types of production we have. Now the transgenic cells, that is the cells containing the rDNA and expressing it, can be cultured in the laboratory. We can use agar plates or suspension culture in shake flasks. And shake flasks, when we talk, shake flasks are used to cultivate the bacteria, fungi, plant cells and the animal cells in suspension. They are easy to use, inexpensive methods because it's quite not, they do not require specific instrumentation and all, and not advanced equipments are required. This method actually needs a flask and a shaker. Actually it's like somewhat like this. When we talk small scale production, it is somewhat like this. The flask is there and it is placed on a shaker. That is, the motor is fitted over here. The shaking allows the mixing up, and it is the orbital motion, and it is for normally very small volume production. So the various small scale includes the shake flask method also, where the very small quantity of the gene product is obtained, so as to it may be for the research purpose or for other reason, but not for commercial use. So the cultures are used then for extracting the desired proteins, and the protein is purified by the use of separation techniques. Now we have in this, in the small scale production, we use the continuous culture system. In the small scale production, we have the continuous culture system. Now what is meant by continuous culture system? It is basically in this method, the medium is actually the medium which is being used over there to cultivate or to allow the host to multiply, that is the medium which is being used, it is drained out from one side of the culture vessel. That means there is continuous removal of the media and the fresh medium is added from the other side. Now the main advantage of this is like whatever medium the culture is being growing on, that medium is removed from one side and the fresh medium is allowed to enter in. The main advantage of this is this actually helps the cell, the growing cell, the cell which is multiplying, to be in their physiologically most active state, and it can be their exponential growth over there. Now this method actually produces large amount of biomass which lead to higher yield of the desired products. So we have small scale production by the continuous culture system where the one medium is like the used medium is continuously being removed and the new medium is continuously being added. As a result, the large scale biomass is produced. Now we use normal, the large scale production. Now it is for the production of the recombinant proteins in large quantities. And for this purpose we use bioreactors. Now the bioreactors, when we talk, there are two types of bioreactors which can be used over here for the large scale production. One is the simple stirred tank, or second is the sparged stirred tank. So we have two bioreactors which are being used for this purpose. Now when we talk of bioreactors, actually bioreactors, as the name suggests, you are providing suitable or ambient conditions for the living organisms to go. That is, bio is the living and making the reaction possible. That is the place. So we talk of bioreactors. Bioreactors are the large vessels in which around 100 to 1000 liters of material can be converted biologically. That is, here we are using the transgenic cells, that is the cells carrying the rDNA or the foreign DNA, into specific products, and we can obtain various products like enzymes, we can have antibiotics, can have growth hormones. So there are various products which can be obtained in the bioreactors. Now the bioreactor provides suitable condition of the pH. So because you know the pH affects a lot. If there is a change in the nature of the solution, it will affect the growth. So pH is maintained over there. The temperature, because temperature denatures the DNA as well as the enzyme. So our optimum temperature is there in the bioreactors. It is continuously being maintained. The oxygen is an important component over here. So the oxygen substrate concentration is also maintained, because substrate has to be there, otherwise no proper reaction will be taking place. Then other factors such as salts, hormones, etc. and the other things, or the other factors, depending upon what kind of product we need, there are various kinds of substances which will be required, and they are maintained optimally in this bioreactors. So let us talk of the simple stirred tank bioreactor. Now before we start with the simple stirred tank, you have to understand that both the type of reactors, bioreactors, have their own disadvantage and the advantage, and it is depending upon what kind of product we need, we can utilize the suitable method. Now the simple stirred tank bioreactor is cylindrical, as you can see over here, and it has a curved base. So the base is curved over here, you can see in the diagram, and it is this curved base provides easy mixing of the contents of the bioreactor. Now it has a system for agitation. That means it can allow the agitator. The agitator helps in even mixing of the oxygen available. Whatever oxygen is available, because the sterile air is being continuously being put in through this. So this is the rotator, and this enables the blade, that is the blade impeller. The blades are continuously rotating, so this allows even mixing of the components. And the foam control is there, so that there is no extra, like it controls or regulates the formation of the foam. Then there is a pH control, that is the acid base controller, or the pH controller is pH is maintained, then temperature is regulated. So there are, and the sampling pots for periodic withdrawal are there, so that for the sample there are continuous monitoring of the product which is being obtained over here. So this method actually involves the use of the blades, and the culture medium is over here. The culture medium is here, and oxygen is continuously being put in. As a result, it is continuously mixed with the help of the impellers, and the pH is regulated. The motor is there which regulates, and from time to time the samples are removed to check the quality of the product. Then we have the second one, which is the sparged stirred tank. It is different from this, that over here in the sparged one, the sterile air bubbles can be sparged. That means bubbles are there which increase the surface area for oxygen, and also the yield can be increased over here. Also you can see the motor which helps in rotating, and this is from the bottom, because the motor is attached over here, so this keeps on rotating, and inside the culture broth is actually mixed up nicely with the help of this motor, and oxygen is sufficient, oxygen is provided so as to have the maximum production. Now when we talk of the two bioreactors, there are two differences regarding the bioreactors, that is the surface area and the oxygen levels. In sparged stirred bioreactor, the vessel is broader. If we see comparatively, the vessel is broader and flat from the bottom. Oxygen is bubbled into the system, and over here we can see the oxygen over here, the upper part. So this increases the oxygen transfer area, and the shape provides increased surface area for O2 transfer. Now, as compared to the sparge stirred bioreactors, simple stirred has less surface area over here, and also there is absence of oxygen bubbles. So this, anyways, both the reactors, bioreactors, are utilized for having large scale production, and definitely sparge stirred tank has a is more is efficient in producing large scale production. Now when we talk of the small scale production, there are also certain advantages and disadvantages to it. The small scale production, the main advantage is the growth occurs at a constant rate, because we are monitoring, it's a small part, so the growth rate, this is, we are talking of the advantages. Growth rate is it is talking about constant rate. Growth rate is at constant rate. Then another important advantage is it is useful for production of primary metabolites. So it is, this method is useful for production of primary metabolites. So the small scale is used over there. Then it is over here, because the culture medium is very small and this growth rate is high, and it is easy to be manipulated. It can be easily manipulated. Easy to manipulate. We are talking of the advantages of the small scale production, but there are certain disadvantages. Now, it is when we are talking of the disadvantages, the contamination can take place very easily over here. Also, large volume of product may be lost due to this contamination. So contamination possibility is quite high in the case of small scale production, and sterile conditions are sometimes difficult to maintain. After, like these two, that is the small scale production or the large scale production, after the product is produced, this product needs to be isolated and then purified. So then it is subjected to downstream processing. Downstream processing refers to a series of processes which are involved in the isolation and the purification of the products and their formulation with suitable preservatives. Why? Because these need to be added, preservatives need to be added for the future use. So if the products are drugs, the clinical trials are required, sterile conditions are required, quality measures are taken care of, and quality testing is also done strictly, which varies from product to product. So downstream processing is an important process after the desired protein or the recombinant protein is obtained.